Thank you very much. Yep, so I'm at the Delft Bioinformatics Lab. I'm an assistant professor there at the Delft University of Technology. And in April this year, we published this paper, Chromatin Landscapes of Retroviral and Transposon Integration Profiles. So I want to talk a little bit about that. And what I'll do at the last few slides, I'll add a few slides on also exploring these long-range interactions between these insertions. All right, so what are we talking about? We're talking about DNA integrating elements that really integrate their little genomes into the genome of a host cell. So these things, they come in various flavors. You have, for instance, the class of retroviruses. You have the murine leukemia virus, for instance, mouse memory tumor virus, HIV, that integrate their DNA, their genome into the DNA of a host cell. And we also have DNA transposons, right, that work through a cut and paste mechanism. And there we have the Sleeping Beauty system and the piggyback system, for instance, that are mostly used. So these systems are really used to, for instance, for gene therapy, where they deliver therapeutic DNA to a patient. But they can also be used to study gene regulation. And I was involved in a study that appeared last year, in which we used Sleeping Beauty transposons to study the effect of local chromatin on expression. And that's a technique called TRIP. I think it's a really cool technique. You can have a look at it. It came out of the Bas van Steensel and Maarten van Lohuizen lab from the Netherlands Cancer Institute. What I'll be focusing on today is mostly related to oncogene discovery in a technique called insertional mutagenesis. So how does this work? Well, these integrations into the genome, obviously, they can be considered as mutations, right? So you get something like this. Now, if these mutations occur in the vicinity of genes, they can activate this gene. And if this gene is an oncogene, this can give rise to uncontrolled proliferation of the cells that harbor that mutation. And in the end, this can give rise to tumors. In a similar fashion, this integration can also occur within the gene, and if that gene is then a tumor suppressor gene, this can also give rise to tumors. Now, we can really exploit this phenomenon by infecting mice with, for instance, a virus, or activating the transposons that are integrated into the genome already. And what you'll get then is that these things start mutating the genome all over, and in the end, this will give rise to a tumor, right? And we can take out the tumor and we can sequence the tumor, and then you get this little integration profile over here. Some of these integrations, they will be driving the tumor, so they are selected for by the tumor development. Others are passenger mutations that basically piggyback on the success of these driver mutations. And one of the questions now is, can we distinguish between these driver mutations that are selected under selection and these passenger mutations? Well, one way to do that is to repeat this experiment many, many times. Right? So you do that in many, many mice, and then you get this aggregate integration profile where these clusters, they point to insertions that are under selective pressure. Right? And these are called common insertion sites, and they're clusters of insertions that when you look in their vicinity, you can identify novel cancer genes, basically. This is frequently used forward genetic screening technique to identify novel cancer genes. Now, back in 2006, we published a method based on Gaussian kernel convolution to identify these clusters. So basically, you convolve the integration signal with a little kernel function, and then you get these peaks that point to these common insertion sites. We came up with a permutation strategy to identify a threshold above which these peaks can be called significant. So then we have a common insertion site that is a region in the genome that harbors more integrations than you would expect based on chance. Okay? Now, there's one assumption in this method, and basically also any other method that you can use for detecting these common insertion sites, and that is that this threshold is based on the assumption of a random distribution, right? But what if these insertions do not integrate randomly? In other words, what if there are biases, right? And actually we know from other work that that is the case. But if that is the case, then these clusters that we observe, we're not really sure that they're due to selection. They can also be due to biases, right? And this can give rise to spurious CISS and therefore also spurious cancer genes. So what we asked was, how do these elements choose their target site in the genome? To really get a grip on this integration bias. And to answer this question, we generated a very large collection of integrations without selective pressure. We did that in 3 systems. The Sleeping Beauty transposon system, where we obtained 122,000 integrations in mouse ES cells. Piggyback system, also in mouse ES cells, 131,000 integrations, covering roughly the whole genome. And the MMTV system in mouse mammary gland cells, and there we obtained 180,000 integrations. So in the remaining slides, I'll mostly talk about the first 2 and compare these a little bit. So now we can use these data to really start characterizing the chromatin landscape of this integration bias. So for this purpose, we collected already published data about 80 genomic and epigenomic features, consisting of, for instance, stuff like gene proximity and the gene structure, DNase 1 hypersensitivity, all kinds of transcription factor binding profiles, and all kinds of histone modifications. And now we use these data to really start generating bias maps, both at the local as well as the global scale. So to give you a quick overview of the procedure that we used for that. So this is one of these integrations, and it has a little bit of signal around it. Suppose that is the DNase 1 hypersensitive signal, but can be any other signal. And then we collected all these signals across all these mutations, and we aligned them. Yeah, it's typical alignment plot kind of thing. And then we looked at the signal at the location of the integration and compared that to the signal at the location some distance d removed from the integration. And we can do a statistical test and measure the association between the signal here and the signal here. And we can do that for various distances, right? So you can really capture different scales in this way. So we can color code the result, and then you get a little bias map for one of these signals for various values of this distance. Now, if you do that for all genomic and epigenomic signals, then you get this figure. It's also in the paper, so if you really want to know which features are here, you should have a look at the paper. So here are the 80 features. These are the scales, and this is the Sleeping Beauty system, and this is the plot we get for the piggyback system. So red is positive association, and blue is a negative association. Now, there's a couple of striking observations that we can make from this plot. The first thing is that there's actually many features for which the systems behave quite similarly, right? So they have the same, approximately the same association for approximately the same scales. The same here. There's also a bunch of features for which there's really major differences. Now, so you can see here that the Sleeping Beauty system, for these features, it had a strong negative association and a piggyback positive association, right? Completely opposite association. There's also some interesting observations to make if we look across the scales. There's a couple of features here and here that are significant only for the somewhat larger scales, right? And these also correspond to the more zonal features, such as proximity to genic features, expression of nearby genes, CPG island proximity, and only a handful of transcription factor and methylation marks. Remaining features are mostly micro features that are significant already at small scales, right? And they include actually most of the transcription factors, DNase 1 hypersensitivity, and most of the methylation marks. Now, by making this distinction, the following picture emerges. So these micro features are determinants of integration bias at small scales, and they're generally relate to the more peaky signals. And we have these macro features, which are determinants of integration bias at large scales, so they're more domain-oriented features. Right? But what's now really interesting to see is that for these micro features, we see strong differences between the two transposons. Whereas for the macro features, most of the stuff is similar. Now, one of the questions that we still wondered about is if these macro features are still influencing the bias, right? Or is everything already explained at the micro feature level, right? So do we really need these macro features? Now, to answer that, we used a Markov blanket analysis where we inferred a Bayesian network for all 80 of the genomic and epigenomic features that we included in our study. And we included an additional node here, the insertion proximity. And then we looked at the Markov blanket, which contains all the nodes on which this insertion proximity depends. Right? So it's conditionally independent of all the other nodes given the Markov blanket. Now we can do that for many bootstraps of the data and train many of these Bayesian networks, and that allows us to estimate a confidence. It's the number of times that we observe a feature in the Markov blanket across all the bootstraps that we did. And we can also measure the association strength with it, which is the average conditional mutual information across all the bootstraps. Now, and what we see now here in blue are the macro features. They're mostly at the top right for both systems, right? So they are very important, and they are selected in the Markov blanket, which shows that they're really relevant here. Moreover, these macro features, they're mostly the same between the two systems, right? Whereas if we look at the red ones, they're also selected in the Markov blanket, but they're mostly dissimilar between the two systems. Now this is then the picture that emerges if you add this all up. Integration site selection is directed at multiple levels. So at a large scale, target site selection is similar across the systems and is shaped by these macro features. And at smaller scales, there are strong differences between the two systems and are observed for actually a wide range of features. Now, is this useful for detecting common insertion sites, right? So how does that impact these results? So what we did is we reanalyzed a large collection of previously published tumor insertion data for 3 systems: the sleeping beauty system, piggyback system, and the mouse memory tumor virus system. And we looked at these common insertion sites, and we counted the number of tumor integrations and the number of unselected integrations that we observed in our screens. And we compared the two, and we did a statistical test to see if there's really more tumor insertions than you would expect based on these unselected integration sites. And we see that actually for all 3 systems, there's quite, in blue, there's quite a few number of common insertion sites that are a little bit dubious, right? So they're potentially spurious common insertion sites, which may lead to also falsely calling cancer genes. And especially for the piggyback system, 33% of these common insertion sites are a little bit questionable if you look and take into account the bias profile. Now, to switch gears a little bit, I also want to tell something about these long-range interactions between these insertion clusters. So far, we have looked at these clusters and we have made one important assumption, and that is that the clustering is on a linear genome, right? But we know to fit 2 meters of genome into the cell nucleus, it needs to be folded up. So it has a 3-dimensional conformation. So what does that mean? So one of the questions that we asked is, are there hotspots that can only be identified when taking this 3-dimensional conformation into account? Right? So you might imagine that there is 2 clusters of insertions that by themselves are not so significant, but if you look at the 3-dimensional conformation, they're actually in proximity to each other, which might give rise to a hotspot and also maybe a novel candidate target gene. Also, it may be that we asked the question, do insertions target genes that are not in their direct genomic vicinity? Right? It might be that through this 3D conformation, actually insertions act on targets that are much further removed than you would expect by chance. And one of the implications is that if you look at a linear genome, you associate the wrong target gene or maybe no target gene at all. So to investigate this, we analyzed a large collection of MLV insertion data that induces leukemia that was published by us before in 2010 and 2008. And that data set was at the time at least one of the largest data set around, and it contains 20,000 integrations in 933 tumors. So the first thing that we did was we clustered the data into insertion clusters, and we obtained 777 of these clusters. But it's important to note that these are not common insertion sites, so we used a much lower threshold, of course, to also include clusters that are not yet common insertion sites. Now, we took the HiC data set that came out of the BinRAN lab, published in Nature in 2012, measured in ES cells, and we asked, so what are now the connections between bins that are underneath this cluster and this cluster, right? So this is the HiC matrix over here, and then these HiC values are located here. And we compared those HiC interactions connecting these two clusters to the HiC interactions of bins that are at approximately the same linear distance in the genome. And that corresponds to these red diagonals over here, right? And we did a Wilcoxon rank sum test to see if that is significant. And if it's significant, we call that a co-located insertion cluster, or a clique. All right, so what does that give us? So if we do that, we identify 874 of these cliques that encompass 479 unique loci throughout the genome. Yeah, they're shown here. 309 of these actually correspond to previously identified CISSes, which is an indication that also these common insertion sites, they actually engage in three-dimensional interactions. 170 of them, they do not overlap with common insertion sites. So they can only be discovered if you take this three-dimensional context into account. Showing you, this is drilling down on one particular example. So this is a locus containing the BRCA2 gene, and it engages in cliques with 5 other loci. Yeah? And what's interesting is that the BRCA2 gene, of course, we know it's implicated in breast cancer and ovarian cancer. But recently, actually, a paper came out that also implicated the BRCA pathway in hematopoietic neoplasms, and including also leukemia. Yeah, so that might give this some positive evidence in literature. It's also interesting to consider the insertion distribution across these 6 loci. So that's shown here. Right, so here are a selection of the tumors, and these are the 6 loci. And one of the striking observations is that there is a really strong pattern of mutual exclusiveness. Right? And this is usually an indication that these insertions really target the same gene. All right, so what have we learned? I've shown you a large collection of data that we can use to find these integration biases. So integration site selection is directed at multiple scales. Globally, we see many similarities, and locally, we see many differences between 2 transposon systems. These integration biases are really important for calling common insertion sites. By reanalyzing 3 large data sets, we actually showed that quite a few of these common insertion sites might be due to this selection bias. And thirdly, we have shown that by taking the 3D chromatin conformation into account, we can reveal additional insertion hotspots and thereby also potential new cancer genes. So with that, I would like to thank some people. So this is Sepide, a PhD student in the lab who did all the work related to the 3D chromatin interactions. This is Johan de Jong. I co-supervise him together with Lodewijk Wessels from the Netherlands Cancer Institute, and he did all the work related to the bias maps. And this is Wasim Akhtar. He is a postdoc in the lab of Maarten van Lohuizen, and he did actually all the wet work and all the generation of the data for the bias maps. So with that, I think I would like to close, and thank you for your attention.
Questions, please, and please use the microphone.
So you're talking about micro sequence biases, but when you say micro, you're talking on the scale of, say, 100 base pairs. For HIV, MLV, and ALV, there's clear sequence bias at a scale of 1 base pair, and the 1 base pair biases seem to actually outweigh the biases that you see, up to 100. Have you examined those single base resolution biases?
Yes, absolutely. So Sleeping Beauty, for instance, only inserts at TA sites, and we were not really interested in that because we know that, right? So what we did, and I kind of left that out in a methodology here, we compared all the associations that we observed to a set of random controls that we chose to only in TA sites. And that allowed us also to correct for other issues, such as the restriction site that was used to clone these things out, and the mappability of the sequencing. So we took that into account.
So just to follow up quickly, the TA site might be a little specific, but for example, with MLV, you see a 10 base region of sequence specificity, and that might be a little more specific than say just a 2 base specificity.
Yeah, so MLV was not included in these bias maps, only MMTV, and there actually we hardly saw any sequence specificity.
Then I would say it would be really cool if you guys could include HIV, MLV, and ALV, because they show that clear, truly microscale bias.
Yeah, that's a good point. Thanks.
All right, other questions? All right, if not, well, thank you very much.
Thank you.
Excellent talk.